Hello, I'm gonna be talking about the weekly physics review today, and I have the information from my notes and from the book. And I also wanted to say that I was recording this is the second video I do. I got interrupted by alarm, so I'm gonna go a little quick. So we're gonna talk about I'm gonna read the instructions as I did first. We're gonna imagine you're working in a research lab, and we're gonna have to understand the transcriptional regulation of G8045, and we're gonna do some experiments to see if P53 increases transcription of G8045 to cells that have undergone the UV damage. So I'm gonna be starting with the siRNA, which is the single interfering RNA, and the goal of this is to reduce the amount of RNA. Reduce the amount of RNA in the cell for the translation of RNA. So the first step is to determine the protein, which is G8045. After we determine the proteins, we look up the sequence of the RNA that can do that. So we can look up sequence of RNA for G8045. For that, third, we buy that sequence, and the sequence is added through translation. So we buy sequence, and sequence is added through translation. After the sequence is added, we wait until 24 to 48 hours of incubation, and after that, we can measure the RNA levels. So basically, we expected the amount of RNA to be less than the amount we started with, but yeah, because of the above processes, transcription increased because they were reducing RNA count. And we're gonna keep going with the second experiment, which is transfection. And the goal of transfection is to bring the RNA in the cell. So the first step of transfection is to buy the G8045 with the control sequence of sRNA and the siRNA. So after we put that all together, we set up the plates because we're in a laboratory after all. And after we set up the plates, we're gonna transfect the cells with the UV damage. After the cells are transfected, we collect the RNA through PCR. And after they've collected the RNA, we're gonna check the cells are in the cell cycle through flow cytometry. So we check the cells in cycle. So let's see where are in the cycle through flow cytometry. So what we expected of this is that we'll allow the siRNA into the cell to make the RNA levels and allow the G8045 to repair the DNA. And the transfection will allow for the isolation to reverse the transcript. With the increased amount of DNA being produced, the cell must divide to compensate for it. So we're going to talk about the third experiment we're going to do, which will be the qPCR, which is the quantification of the PCR. And the goal of the qPCR is to reverse the transcription to reproduce DNA out of RNA. We're going to reverse transcription from RNA to DNA. Wait. We're going to produce DNA from RNA. So For the first step, we're gonna isolate the DNA. I have to isolate the DNA, and then we convert the RNA to DNA. And how do we do that? We're gonna qPCR with a CyberGreen, which I think it's a protein. And after we do the qPCR, the computer will control the fluorescence of the experiment and the cycle of quantification, which is so the computer we're measuring the PCR on will measure the fluorescence. Which is the CQ, I'm sorry, which is the cycle of quantification. So this way we can see the fluorescence of the DNA. We're going to expect that we allow the new DNA to be made of the RNA cell, and the new DNA produced can't count like the DNA, new DNA is produced and the count of DNA is also increased. Now for the last experiment we're going to do in the laboratory answering these questions, it will be the flow cytometry. So for the flow cytometry, the goal of the flow cytometry is to assess where the cells are within the cycle. So the first step of this is to wash with the PBS, and we're gonna permeabilize the cells. Wash with PBS, which we have used a lot. And we're gonna permeabilize cells. After we wash with PBS and cells are permeabilized, we can stain with propodium iodide in the presence of RNase, which is the enzyme for RNA. And this will stain the DNA, or it would degrade the DNA. So after we stain with the propodium iodide and in the presence of the RNase, it will stain the DNA, or it will degrade it. Either or. And for what we expect, we expected that the flow cytometry to allow us to know the status of the cells during the DNA being repaired from the UV exposure and the damage. These aligns with the job, the data showed that the P53 increased transcription of G8845 because the UV light caused damage to the DNA, where G8845 job is to repair the DNA. And these processes helped us to investigate that, and this aligns with the job with the being controlled cell division. And that's it. Thank you.